everyone. My name is Erin Baker, and I'm an associate professor at North Carolina State University. I'm really excited to show you today some of the work my group is doing with multidimensional analyses in exposure studies. So let's get started. So my research interests have always been on a clinical side of why is a person sick, how should the person be treated, and can we detect diseases before a person presents symptoms or becomes symptomatic? So a lot of times we take biofluids or tissues and we look for metabolites, proteins, or lipids in those biofluids that could be specific markers for diseases. Recently, we've also been incorporating different xenobiotics, which are molecules that get into your body through either environmental exposures, such as pesticides on foods or pollutants in the air, and how those actually change your biomolecules. And then we really want to make sure that we have good visualizations of these because you can get a lot of data from these analyses. So that's another area my group works on, but we're not going to talk about that today. So before I get started, I always have to thank the people that did all the hard work and then let me present it. So these are the students in my group, and they're working on this comprehensive molecular profiling to have a better understanding of systems perturbations and doing all kinds of different omic analyses, such as these exposed omic analyses for the xenobiotics, all the way down to metabolomic analyses for those endogenous metabolites. So why we're really interested in this project is when the Human Genome Project was finished, it illustrated that greater than 90% of diseases are not due solely to genetics. And this was done through studies of monozygotic twins where they saw that they didn't die in the same way. And there had to be some environmental exposure that was causing these people to actually have different ends of their lives or ways that they died. And so what we want to do is we want to look at these external environmental exposures, such as what you eat, your diet, your lifestyle, the pollutants you're exposed to, the drugs that you might be taking, and the cosmetics that you put on your face and use in your shampoos and your soaps, and how those get processed through your body, because unfortunately everything is sucked either through dermal absorption or through your mouth or through your nose as you breathe in these different molecules. And we see that those can affect your xenobiotic metabolism as they go through. They can change and have different properties as they change. It can change your endogenous metabolites, and then it can also change your gut microbiota, all resulting in measurements that are commonly known as just metabolomic measurements because all of these molecules are coming together through your system. However, we know that it's not just the metabolome, because if we look at those specific molecules in the KEGG pathway, some of them are glycans, some of them are lipids, and there's those proteins or enzymes that are causing these different changes to occur. So doing a multi-omic evaluation of how these xenobiotics are processing through your body is very important. And what we call this is, if you're looking at specifically the xenobiotics and its transformation, those are direct measurements of exposure. Well, if we're looking at these other omic analyses, those are indirect measurements because that's seeing how the other molecules are affected by the exposure. So many people for these studies use liquid chromatography and mass spectrometry or even gas chromatography for these evaluations. But our group wants to do even more thorough assessments of these different molecules, and so we incorporate a third dimension into our analytical measurements. And we call this ion mobility spectrometry, or it's called ion mobility spectrometry. We don't specifically call it that. But what it allows us to do is it's a rapid gas phase separation of molecules that gives structural information. So we can put it right after the liquid chromatography separation that gives us information on hydrophobicity and hydrophilicity. And then right before the mass spec evaluation that gives us mass information, so we can get 3 dimensions for every molecule that we detect at our detector. And the specific ion mobility spectrometry analysis we use is drift tube-based ion mobility spectrometry. And in drift tube-based, we have a tube that's full of an inert gas, and we like to use nitrogen for that because it's renewable. Some people like to use helium. And then nitrogen is also more cost-effective. And what we do is we put about 4 tar nitrogen gas in our drift tube and then apply a very weak electric field so that we can pull the molecules through that drift tube and allow them to separate by different sizes, because the smaller molecules are going to have fewer collisions with the nitrogen gas than bigger, more extended molecules that'll collide a lot more as they go through this tube full of gas. So for an example, if we have 2 different peptides here, and that one has a more folded shape and one has a more extended shape, if we pulse those, because they have to have a start time and an end time, it's basically a race through that drift tube. If we pulse these peptides into the drift tube, we're going to sit there and watch and see which ones come out first. And in this case, both of these peptides have the exact same mass, or M/Z, so they would have the exact same information at the mass spectrometer. So here we can see that the more folded compact one makes it through the drift tube faster than the more extended form, and we get separation based on these 2 different shapes of the molecules. And it tells us that the more compact one, there's about 40% of it in our sample, and the more extended one, there's about 60% of it based on the peak height. So these ion mobility analyses allow us to get an ion neutral collision cross-section. And that why we call it an ion neutral is the ion is what you're looking at, and the neutral is that buffer gas or nitrogen that's surrounding your molecule. And it's basically making it look like a sphere on the right, because it gives it a surface area where if you change out your buffer gas, that surface area is going to go up or down depending on the different gas sizes. But the nice thing about collision cross-sections are they can be easily compared between different labs because they normalize out the length of your tube, the pressure, and the temperature of your measurement. And so they're just looking at that specific velocity that the molecules move through the drift tube with. And so this allows us to share our SOPs with labs in Australia or Europe, and it allows other people not to have to run every single molecule we do. They can just take our collision cross-section values and compare that to their experimental measurements. And so by having LC first, then ion mobility second, and then MS third, all of these separations are done simultaneously, but we have to make sure that our each one is faster than the next so that you're sampling the peaks as they're coming out. So our LC is going to have peaks in seconds usually, and the LC separations are going to take place in minutes, where our ion mobility analysis time is normally about 60 microseconds. So it's sampling those second peaks in a millisecond time scale. And then we use time of flight because it has the rapid pulsing. And so then we can analyze each of that 60 milliseconds in about 150 microsecond pulses. So ion mobility is very fast. It can be coupled with these other separations. It allows for multidimensional analyses for more confident identifications, because if you're looking at a library, now you have 3 different dimensions to match your library to. And these collision cross-section values allow us to take potential identifications and see which ones are actually the closest in structure and eliminate some that might be more extended than what we've seen in our experiment. We can also use it for interference removal, so it increases our confidence. So here we can see this is our LC-TIC. So if we look at a specific peak here at around 22 minutes, and then if we look at our ion mobility and our mass spec information, we can see our ion mobility is on the y-axis. That's showing how fast our molecules drift through the system. And our mass information is on the x-axis. And at that specific time of 22 minutes, we see there's quite a few molecules. And in the mass spec at the very top, you can see I have dotted lines for the molecules that I'm interested in actually observing. And we can see there's quite a bit of interference in those first 3 peaks. However, if we slice out the drift time that we want, this is including all of those different molecules and the different drift times. If I slice that out, I can clean up all that other interference or molecules that I'm not completely interested in. And you can see I've basically removed any noise and other molecular interferences and have a very clean spectra for quantitation of those molecules that I would like to see. The other thing ion mobility is very good for is it's good at showing molecular trend lines or relationships. And this has been shown for years. This is a paper from John McLean's group in Vanderbilt, where in 2008, they showed that using ion mobility, we can get different trend lines if we plot the M over Z of the molecules versus their structure. And we see that lipids have a very high slope, where nucleotides and carbohydrates have a lower slope. And the reason for this is per M over Z, the nucleotides and carbohydrates are very ring-based, so their atoms are bound up tightly. That allows them to go through the drift cell a lot faster, so they have a lower collision cross-section than lipids, which are much more extended. This property is very powerful, though, because it helps us look at other molecule types and see how they're separating. So here we did the same thing as we took some oligosaccharides, peptides, nucleotides, and lipids, and we saw the same trend line, and we could start discriminating them based on their different M over Z versus CCS relationships. But then what we wanted to do is we wanted to take the small molecule area and take as many standards as we could find and see if we could see specific relationships for the small molecules we wanted to study. So here we started looking at some of the trends, and it was very cool because we were able to see these specific relationships. And here you can see bioacids, because they have that steroid type backbone and just modifications of the hydroxyl number and position, have a very flat slope and a different relationship than our nucleotides, our nucleosides, and then our lipids, which are, again, our most extended, which also include our fatty acyls now. But we really noticed that our xenobiotics, such as our polybrominated diphenyl ethers, had a very low slope, and it was even lower than our very ring-based biomolecules, which we thought was very interesting, as now we've added halogens to our molecular or our aromatic rings, and we're seeing completely different slopes. So we started to look into this a little bit more, and we looked at PPDs, and also still having those PBDEs in there, we could see that we basically had an exposomic realm where we were separating our biomolecules, which are seen more up high in this plot, from our exposure or our xenobiotic molecules. And we thought this was really interesting, so we wanted to really look into that and see how true that was. So some molecules that are of great interest right now, and it should have been for a long time, but are really being brought to the public's knowledge, are these poly and perfluoroalkyl substances, or PFAS. And so PFAS are fluorinated aliphatics, so that's basically taking that hydrocarbon chain and putting fluorines for every hydrogen. And this allows them to be chemically inert, thermally stable, and hydrophobic. And what those properties enable is that allows you to basically not have them bind with water or with grease. And so people like to use these in stain stick and in raincoats and Gore-Tex material to keep the water and the grease away from the different materials. The other thing that they're used in is in your food wrappers. So the bottom portion here where it gives the percentage is the percent of fluorine in these different wrappers. So you'll see them in your cookie wrappers, your burger containers, your French fry wrappers, because that keeps the grease from seeping through onto your hands from these different foods. But luckily we don't see them in our paper cups, because the scary thing about these is they're noticing that they don't process through people. They actually bioaccumulate in your systems and in your organs. And as you get more and more exposure through time, they don't clear. They just keep adding up and adding up and adding up in the system. You guys might also have heard about Teflon pans. PFAS are one of the main components of Teflon pans. And they are in carpets and furniture for those stain sticks, so that when you drop that grape juice on your carpet, it doesn't seep through and leave a big stain. You can go wipe it up really quickly. And then they're also included in many firefighting foams. And so they are known in AFFFs, which are aqueous film forming foams. And this helps put the fires out. But PFAS are becoming a global contamination issue. We're seeing them all over. We're actually seeing them in the blood of polar bears, because once they get in the water stream, they don't degrade. They're we've seen PFAS in some of our systems for over 80 years, and they were just made in the 40s and 50s. So I think sorry, over 70 years. So unfortunately, they are very, very robust and don't like to break down. The other bad qualities are they're being linked to increased thyroid disease risk, increased cancer risk, developmental effects, liver damage, immune system suppression, and increased cholesterol in different people. So they're very frightening because they're having these horrible effects on people with direct linkages in many cases. So in North Carolina, we're specifically interested in these because one of the chemical companies that make PFAS is in North Carolina. So we wanted to see how much contamination we were actually seeing in our environment, and I'll talk a little bit about that later in this talk. So first, what we want to do is there's many different types of PFAS, and the EPA gives you numbers anywhere from 5,000 to usually up to 9,000 different PFAS species that could be occurring due to products that are made, and then degradation of some, metabolism of others, and then just different ways that things are being made for these PFAS. So if we looked at the ones on the far right, so these are the PFCAs, or the carboxylic acid head groups. You can see there's different head groups on these four subclasses that I'm just noting here. The PFCAs are the most similar to our fatty acyl groups because fatty acyls have a carboxylic head group and just have hydrogens where all of these fluorines are in the PFAS. So we wanted to see in our M/Z versus CCS trend lines, are we seeing a difference between lipids and these PFCAs? So we started by looking at different versions of the PFCAs because they're basically polymeric. They have different numbers of CH2 groups. And when we looked at them, we saw that we could see either a deprotonated form where we just lost the hydrogen on that carboxylic acid end, or we lost the whole carboxylic acid. So that also caused another ion. So you can see the two different trend lines for the two different versions we see in our instrument. However, if we compared these to the lipids, we see a huge separation in these trend lines of the lipids versus these PFCAs, which was very good for us because lipids are commonly extracted with PFAS. So we want to know that we're confidently identifying the PFAS and we're confidently identifying the lipids, and that we're not getting confused by either one of these species in our complex systems if we're looking at blood or if we're looking at plant tissue. And the main reason for this difference, because you might think these guys are very similar, but it's because fluorines have a mass of 19. So that moves them up in the M/Z x-axis, but the fluorine doesn't have that much difference in size than a hydrogen. And so we see that if you look at that spot around M/Z of 200, for similar types of classes, we see for the PFCAs that we just have 4 carbons and 9 fluorines match something that has 12 carbons, 23 hydrogens, and 2 oxygens for a lipid species, because those hydrogens are only adding 1 Dalton. So you have a lot longer lipid, which is going to have a bigger cross-section than our shorter PFAS, but just because of that change in M/Z of those halogenated compounds, we're seeing that big shift. So then we started looking at the different subclasses, and we could actually see differences in the subclasses of the PFAS. So if we were looking for specific contamination sites, we could actually tell you what subclass the molecules were coming from. And so this made us really excited. The other thing is we could see differences in things such as 6,2-FTS and hydroEve that can commonly be put together or combined when some people are doing these analyses, because they often elute together in the LC and they're very close in mass. So sometimes it's very difficult to tell them apart, but we see huge cross-section differences, and those are shown by the 2 asterisks, the pink and the yellow asterisks on this plot. So then what we want to do is we said that we think we have a really good method for looking at these different subclasses of PFAS. We need to compare this to the golden standard, which is a triple quad mass spec hooked up with LC separation on the front end. And so we used this specific method that is commonly used by many doing PFAS analyses. And then we went and we got some wastewater. We took that sample and we used SPE to concentrate it, because a lot of times you want to get a decent amount of water, because the PFAS hopefully isn't going to be screaming high, but there's still a decent amount of PFAS in a lot of the water systems. And then we used our LC-IMS-MS mass spec platform to analyze our water sample, and used our library that I was just showing you with the M/Z and the CCS values to actually see what PFAS we were detecting in the water samples. So it was really cool, because we actually saw 6 different PFAS, and we also saw 6 different PFAS in the triple quad. So we had a direct validation that our method and the triple quad were giving us the same answers. And if we look at our wastewater cross sections for these PFAS that we detected, and then we go to those standard cross sections, which were our values in the library, our percent error was in no cases bigger than 0.3%. So we were very confident with these identifications. We also used the LC retention time, the mass spec information, and the MS-MS information for further confidence in each of these. So this multidimensional information was so important for our exact identifications of these. So now if we go back to our charts of our M/Z versus CCS, we wanted to put these PFAS in that same area with all these other biomolecules that we've analyzed for our libraries. And so I've shown before, the PFAS are lower than lipids, but how do they relate to nucleotides or bile acids or some of those other molecules that are commonly detected in negative ion mode? So here's our PFCAs, and this gives kind of an average value for the PFAS that we're analyzing. So I didn't put them all on there, but just for clarity, it's more the PFCAs. Then if we add all of the other molecule types that we've evaluated in our library, we can see that those PFAS are way different than all of our biomolecules. So we're getting huge separation because of that increase in the mass for the fluorines and the difference with those molecules that are heavily hydrogenated. And then if we add our PBDEs and our PCBs in there, we can also see that we get this exposomic realm again, where we have our halogenated exposomic molecules, but our PFAS, interestingly, have a lower slope than almost all of our other molecules, other than the PBDEs at the very high M/Z when we start adding lots and lots and lots of bromine to those PBDEs. We also see that the PCBs are kind of in between our biological space and our halogenated realm until we start adding more and more chlorines. We can see them drop from the in-between space into that halogenated space because they have more effect of the chlorine ions. And if we look at our PAHs specifically, those do lie in between both of these spaces in these different cases because they don't have the halogenated molecules in them. And so what we wanted to do with this information is we wanted to do some environmental monitoring. And we were specifically looking at different things that are being done. And so the EPA has methods to look at surface water and groundwater. And then the NCDEQ has air quality sampling where they look at rainwater. And then the EPA has methods for soil and sediment. But one area is the atmospheric transfer of PFAS, which people are thinking is extremely important to understand because anytime that wind blows, where does the PFAS end up? So we wanted to look specifically at the trees and see how much PFAS actually gets stuck in the trees that are near contaminated sources. And so we thought that we would like to use pine needles as passive samplers for these PFAS. And the reason we thought about pine needles is some work had been done on a few species of PFAS. And what they've seen is that pine needles have very waxy needles. And the wax likes to bind up the contaminants in the air. So they've used them for pesticides, PCBs, PAHs, PBDEs, metals, and a few PFAS. So because we have this extensive library now, we wanted to see if we could go further than just seeing a few PFAS, could we see more? And so this is some of the existing data where we can see that in Europe and China, they were looking at these legacy PFAS. And why they wanted to use the pine needles on the far left is that they don't have as much seasonal dependence either as some of these other trees, such as poplars or willows. And they had the most amount of the PFAS in them. So they were able to retain the most in those needles. So we wanted to see, can we actually use these pine needles for analyzing legacy and emerging? And the emerging haven't been seen, very few emerging have been seen in the pine needles previously. So we started sampling at different locations throughout North Carolina, and specifically around the plant that's known to make PFAS, which is the Chemours plant. And we sampled pine needles from 2018 and 2020. And then this is where we started evaluating those different needles. And here you can see why it's so important to use that ion mobility dimension, which is on the y-axis, is our biomolecules have a completely different line than our PFAS molecule. And the PFAS molecule is highlighted by the pink bar. Here we can see that that 4,2-FTS is very much separated by that fatty acyl 22,6. And you can see the fatty acyl structure on the top and the 4,2-FTS structure right above the purple line, and how different they are. So the structural separation of the ion mobility was huge for separating them, but the masses are very similar. So M over D, you might get some interference if you're just doing mass spec alone. And so using our library for our PFAS, our library contains over 100 different PFAS that we've specifically run with LC, ion mobility, and MS-MS. And we were able to see over 70 different PFAS in the pine trees throughout North Carolina. So not all in the same trees, but through different distributions. And so these covered all the different class, or many different class types, so different head groups and different chain length types. And previous studies where the best we could find have only seen that small portion on the bottom right, where you can see that for they have the best coverage for the PFCAs, which are mainly some of your legacy PFAS, but they had very little coverage for many of the other classes or subclasses of PFAS. And so how we wanted to visualize where we were actually seeing these PFAS was by doing toxicological prioritization index plots, or ToxPis. And in a ToxPi, what you do is you look at all of the sites you're interested in and basically normalize each PFAS that you're detecting to the highest site. So the highest site PFAS will have a value of 1. If it's not detected, it'll have a value of 0. And that's illustrated by the height of the slice in your ToxPi. So here we can see PFBA would have a 1 value at this location, so it would be very high. It would be the highest in all of our studies, where PFBS would be very low. It would be detected, but it would be one of the lower values in the study. And so this shows the slice and how 0 is not detected and 1 is your max abundance. But this gives us a good way to look at distributions. So what we can do is we can take these ToxPis now and we can put them on maps and see specific trends in the distributions of the PFAS we're observing. So you can see that pi in the middle with all the PFAS names around it, that would be the max for all cases. So that's not at any of the sites, but that would be the max for all cases where you have a value of 1. But now if we start going by our chemical plant, which is shown by our little cartoon industry building there, we can see that we're getting very high values for a lot of these PFAS that we're specifically looking at. And here we're looking specifically at the fluoroether PFAS. If we go to an airport, we're also seeing high values of certain ones, but a lot of not detects for the ones that we were seeing from that chemical plant. And then if we go down to the ocean, we're also seeing one PFAS that looks like it's specific to areas around that ocean water. But these are all specifically for those pine needles. So all this sampling is done for the pine needles. Then if we go to another airport, so this is the Raleigh-Durham International Airport, we see a different distribution of PFAS. And now we actually saw for these PFCAs and PFSAs that they were 2 orders of magnitude higher than any of the other sites in North Carolina that we were looking at. And we specifically found a cyclic PFAS, which is on the top left, that is related to hydraulic fluid. And so the interesting thing is that the North Carolina National Guard is located right there. And we were seeing the most contamination in that area, which we were guessing was due to testing of potential firefighting foams, or those AFFFs that have high levels of PFAS. So since we were seeing these molecules in these pine needles, we wanted to go and check historical archives. And so we went to a herbaria in both North Carolina State and then one at Duke, and got pine needle samples from the 60s to the early 2000s to see if we could see how these PFAS were changing in North Carolina. And what we saw here is, if we look at our different timelines, so we have our timeline on the x-axis and then our location in North Carolina on the y-axis, with different counties in North Carolina highlighted by the different dashed lines, we can see that we saw in the early 1960s very few PFAS, but we saw an increase in the number of PFAS as we went through time. And some of the main trends we saw is, if we looked before 1985, we saw usually fewer than 8 PFAS detected, and after that we saw an average of 14 PFAS detected in the different trees. So we also noticed that 1967 was a little bit of an outlier in our early testing times, and we found out this site was right next to a military base that was using AFFF. So we can see that that location versus the other location that was more in southeastern North Carolina is quite distinct due to that military base presence. This historical mapping really helped us learn a lot about the PFAS. So now we're specifically looking at lipid analyses of different things, specifically plants and animals that might have PFAS exposure, due to the direct correlation they've seen with lipids and PFAS and the similarities in structures. So there's been links to increased cholesterol levels among exposed populations, higher risk of hypertriglyceridemia, and then a partitioning into bacterial cells and membranes, and then also suppression of genes involved in fatty acyl metabolism. So this is an area that hopefully you'll hear me talk about in the future. And so I hope I was able to show you in this talk that with ion mobility, we were able to remove interferences, increase omic ID confidence, and enable rapid analysis with this multidimensional information. We also use these molecular libraries for experimental CCS, and other people could use those for theoretical. And then I wasn't able to show you our work with our visualization and processing of the big data. But hopefully we can take our little guy here who's got his telescope pointed at the wrong direction for looking at the comet, and give him more information and have him start looking in the right direction. So a big thanks to my group, all my funding sources, and all our amazing collaborators for all their help with this work. And I hope you've enjoyed this talk. Thank you so much for tuning in.